What's going on, guys? It's Josh with Rise Above, and I'm back again today with an update on my experiment to boost memory as laid out by Leo and Longevity on his channel. Before we get started, please be sure to subscribe to the channel and like the video. Your support is very much appreciated as I look to gain further attention for this platform. As you can see, I'm slowly upgrading the little studio I have going on here. Progress over perfection, right? But enough about that. Let's get into the video. Today is an update on my biohacking experiment to increase cognition and boost memory retention and synthesis by stimulating the cholinergic system. And of course, all credit due to Leo and Longevity, who I picked this advice up from. But here's the background. By stimulating the cholinergic system, I hope to further improve my memory, and this can be done in a few ways. Choline, the base nutrient that provides fuel to the cholinergic system, is a precursor for a neurotransmitter known as acetylcholine. Acetylcholine is the primary neurotransmitter associated with memory storage and synthesis. The theory goes that by increasing choline uptake with supplementation and by adding this on top of existing therapies for Alzheimer's patients, one should be able to augment and improve physical memory retention and the ability to remember things. We do also make choline in the liver, but not in amounts sufficient to meet requirements of the body, so dietary choline is required. Choline is typically found in eggs, various meats including beef, fish, chicken, and livers, cheese, as well as in broccoli and cauliflower. To continue the background, let me show you how your dietary choline factors into your brain's ability to process and remember things. This right here is an image of a neuron, which is a type of brain cell responsible for various types of communication. The neuron has something called an axon, which extends out, and each terminal, as they're called, at the end of the axon, comes in very close proximity with the terminal at the end of the other neuron it's communicating with. That segment is called the dendrite of a neuron that comes into that close proximity area with what's called the presynaptic neuron. The endpoints of the axon are referred to as presynaptic terminals, while the dendritic portion of the other neuron are called postsynaptic terminals. Acetylcholine is created from an interaction between acetyl-CoA and choline, the nutrient I'm talking about, which are acted on by enzymes called choline acetyltransferase. The acetylcholine is synthesized from this reaction and then is stored in basically these little sacs or vesicles inside of that presynaptic neuron. The stored acetylcholine is then released under certain conditions, then transmitted across the gap between neurons, also known as the synaptic cleft, thereby leading to a communication between those two neurons. Of course, this happens with a myriad of other neurotransmitters as well, but in this case, we're just talking about acetylcholine. So if you're deficient in choline, then you can underproduce this key neurotransmitter, leading to a reduced transmission of that acetylcholine across the synapse. Patients with Alzheimer's tend to have neuronal degradation and often have low levels of acetylcholine in the brain. Now for the types of choline. The popular and primary forms of choline that can be purchased for supplementation are CDP choline, PC choline, and alpha GPC. Each of these play a slightly different role in the choline system, either implicated in cell integrity, storage of choline for later release, or breakdown for release of more choline for acetylcholine formation. In this image, you can see the different paths that choline can take after being ingested. The first yellow CHOL is a choline molecule, which can either go left in the diagram and be transformed into acetylcholine, or head downwards and then turn into phosphocholine, and then into CDP choline, and then into quote-unquote PC choline. The conversion of choline into PC choline is one half of what is called the Kennedy pathway. It's not super important for our purposes, but just in case you guys wanted to know the name. CDP choline is a middleman in the synthesis between regular choline and PC choline. And PC choline is the one that is stored in cell membranes and assists with cellular integrity. According to medical journals, CDP choline breaks down into both regular choline and something called cytidine on oral ingestion. Ingestion of CDP choline can also increase levels of dopamine and norepinephrine, as well as eventually the levels of acetylcholine that we're looking for. Studies suggest that CDP choline supplements also increase dopamine receptor densities. meaning increases the amount of dopamine receptors in the brain. The choline system is tied to the dopaminergic system in that way and can somewhat enhance dopaminergic activity. Alpha GPC, this type of choline is essentially particularly effective at crossing the blood-brain barrier and contains more choline by weight than CDP choline does, and also breaks down into choline and cytidine when administered orally. It leads to an elevation of choline in the brain, particularly an acute spike of choline there within 1 to 3 hours of ingestion. It does lead to overall elevated levels, but particularly there's that spike in that first 1 to 3 hours, which is why it is suggested you take it in the morning. So alpha GPC is a naturally occurring constituent of red meat products and organ tissues, like I mentioned, but for the most part is scarce in appreciable amounts in naturally occurring sources. Supplements containing alpha GPC are made synthetically, either from eggs or soy lecithin. And PC choline is a type of choline which is a major component in the cell membrane in just about every cell in your body. That's how you can sort of supplement those things. So to recap my plans here, let's review the first part of my intervention, which I've already started, the cholinesterase inhibitor. To assist with improving my memory, I'm currently taking something called Donepezil, a cholinesterase inhibitor specific to the central nervous system. The active compounds in Donepezil, among other similar drugs like, I always have trouble saying this, forgive me, rivastigmine, I believe, could be saying that wrong, bind to various sites on the acetylcholinesterase enzyme in order to prevent or reduce the amount of breaking down of acetylcholine that it does. This leads to more acetylcholine remaining active at that synaptic cleft or floating in the space between neurons for longer periods of time where it can continue to perform its action. The purpose of adding additional supplementary choline will be to enhance the effect of Donepezil and the inhibition of that acetylcholinesterase enzyme, essentially adding more fuel or gasoline onto that proverbial fire, so to speak. So besides expert advice, what research did I find to back this up? And what does it say about what can happen if I do? Is this just mad science? Not really. I found some very interesting results, so let me show you the research I found to back this up. For the first type of choline, the CDP choline, a 2002 review entitled Neuroprotective Effects of CDP Choline on Impaired Spatial Memory in a Rat Model. Essentially, it shows in the rat model, the rats were better able to navigate the maze, reduced incorrect choices in the maze, and increased correct ones exponentially along with the increasing doses of CDP choline. Next, a 2002 review entitled Clinical Reviews of Cholinomimetic Agents. So for background, citicholine, or CDP choline has been approved in Japan and Europe for traumatic brain injuries, stroke, and other neurological disorders. Several small-scale clinical trials have demonstrated the efficacy of CDP choline in traumatic brain injury. In a study by Levin in 1991, 14 subjects were treated for post-concussive symptoms after mild to moderate closed head injury. Improvements were noted in recognition memory, but also in symptoms such as headache, dizziness, and tinnitus. Other trials have shown decreased length of coma, decreased length of hospitalization, improved motor recovery, and even enhanced rehabilitation potential in the CDP treatment groups. And next, a systematic review of human models in Alzheimer's utilizing CDP choline. A 2020 review entitled Effects of CDP choline as an Adjunct Treatment for Alzheimer's Disease. Quote, the purpose of this study was to determine the effects of cholinesterase or CDP choline as an adjunct therapy to cholinesterase inhibitors in the treatment of Alzheimer's. 2 retrospective cohort studies involving 563 elderly patients affected with Alzheimer's were included. After 3 months and 6 months, the intervals of which they measured, better mini mental state status examination scores were observed in the cholinesterase inhibitor plus CDP choline group versus cholinesterase inhibitors alone group. The adverse effects of combined treatment were self-limiting and included occasional excitability, gastric intolerance, and headache. The conclusion was it's well tolerated and showed an improvement in cognition, mood, and behavioral symptoms compared to cholinesterase inhibitors alone. Finally, a 27 multi-center study entitled CDP choline plus cholinesterase inhibitors in aged patients affected with Alzheimer's disease. The aim of the present study was to show the effectiveness of oral acetylcholine plus cholinesterase inhibitors in patients with Alzheimer's disease. It was a retrospective, multi-center case-controlled study, including 7 centers for cognitive impairment and dementia in Italy, with 448 consecutive patients aged 65 or older with Alzheimer's. 197 patients were treated with a cholinesterase inhibitor, while 251 were treated with cholinesterase inhibitor plus 1,000 milligrams of CDP choline orally every day. Cognitive functions were assessed by mini mental state exam, as well as daily life functions, behavioral symptoms, comorbidities, and mood tests. Patients treated with CDP plus cholinesterase inhibitor showed a statistically significant increase in mini mental state exam between the 3-month and 9-month period. Here's a diagram showing the effects of cholinesterase inhibitors plus CDP on the mini mental state baseline, 3 months and 9 months. These tests typically involve things like word recall, number sequencing, things like this. There's even a part where you fold a piece of paper and pick it up off the floor to test your physical ability. Next, alpha-GPC, the other type of choline I'll be supplementing with. For alpha-GPC, I found the following studies and reviews. A 2013 review entitled Choline Alpha-Cerate, an old choline-containing phospholipid with a still interesting profile, which stated that, quote, choline alpha-cerate or alpha-GPC has significant effects on cognitive function with a good safety profile and tolerability. Although limited both in terms of size, the samples investigated and the clinical length of treatment, preclinical and clinical results presented suggest that cognitive enhancing capabilities of choline alpha-cerate merit being a further investigation in appropriate trials. Next, a very big important trial in this area, which was initially a 1-year trial, but extended to 4 years based on favorable results. From 2020, quote, interim 3 years results from the ASCO-MALVA trial. ASCO-MALVA trial is a double-blind trial where patients are assigned to Donepezil plus placebo or Donepezil plus alpha-GPC treatments, and then underwent brain magnetic resonance imaging, or MRIs, and neuropsychological tests every year for 4 years. An interim analysis of the 3-year MRI data was performed. Two groups were compared: a treatment with Donepezil at 10 milligrams per day plus alpha-GPC at 1,200 milligrams per day, and Group B was treated with Donepezil 10 milligrams per day plus a placebo. In Alzheimer's disease, it is characterized by decrease in the weight and volume of the brain. The Donepezil plus placebo group developed atrophy of the gray and white matter in the brain, and in the Donepezil plus alpha-GPC group, that atrophy was less pronounced in frontal and temporal lobes, in the hippocampus, and the amygdala. The morphological data presented here is consistent with the results of the neuropsychological test as well. Figure 3 here shows segmentation of the cerebral areas obtained by MRI analysis. And this analysis enabled extrapolation of the volumes of the gray matter, white matter, and cerebral spinal fluid. Interestingly, the cerebral spinal fluid appears to increase as compensation for the loss of gray and white matter. Next, figure 4 of this study shows the results of volume analysis. A progressive reduction is noted in the volume of the hippocampus, and it was noticeable along the course of the entire study. The reduction was again more pronounced in the Donepezil plus placebo group than in the Donepezil plus alpha-GPC group. The difference between the two groups was significant for the right hippocampus throughout the 3 years of treatment, whereas the significance was recorded in the 2nd and 3rd year of treatment for the left hippocampus. Cognitive assessment showed a significant difference between the two groups. Specifically, the Donepezil plus placebo group showed a significant worsening of the global cognitive functions measured through the mini mental state exam, as well as the ADAS cognition test, compared to the group with Donepezil and alpha-GPC, and this was seen at both the 2-year mark and the 3-year mark follow-up. Results from the 2nd year of the ASCO-MALVA trial indicated, again, a significant decrease of severity with Donepezil plus alpha-GPC compared to Donepezil with placebo. Mood disorders such as depression, anxiety, and apathy decreased in patients treated with Donepezil and alpha-GPC, while the severity and frequency was increased in the other group. The authors wanted to state as well, the study has strengths and weaknesses, while the size of the sample and nature of the post-hoc type of analysis are weaknesses. So they do state a larger clinical trial would be needed to further determine efficacy and advantages. For the actual plan for the treatment, my intervention that I'm going to be doing, based on these trials, although not humongous in size, the good safety profile of these supplements, I will be starting to add to my current regimen of 5 milligrams of Donepezil. I will be adding 250 milligrams per day of CDP choline, adding an additional 250 milligrams every 3 days until I reach about 1250 milligrams. I'll be testing my reaction, mood, and cognition along the way, and I will be splitting any additional intake after the 500 to 600 milligram mark into later portions of the day across meals. From there, I will perhaps wait a week to see if the results stabilize or provide some sort of major difference. And then I will apply the same strategy, adding 150 milligrams of alpha-GPC, increasing by 150 milligrams every 3 to 4 days until either 600 or 1200 milligrams, depending on my response. That will all be taken in the morning of the alpha-GPC. After that, I will evaluate the results and decide whether to conduct a washout period, meaning a reset period, with the choline supplements, so just completely remove those, and then do the entire experiment again at 10 milligrams of Donepezil. So these supplements are generally considered safe, and although dietary choline intake is recommended at 200 milligrams per day by the NIH, the Linus Pauling Institute indicates that adults should not consume more than 3.5 grams of choline daily, and the tolerable upper limits for adolescents and children are slightly lower at 3 and 2 grams respectively. These are both over-the-counter supplements, so there's a little concern in the way of safety. In conclusion, that's it for now. I have a melatonin biohacking video in the works as well as a follow-up to my last video, and I will likely post that next. For now, take it easy, and I'll see you on the next one. Thanks, guys.